The overall goal of this procedure is to measure the levels of heme synthesis in mammalian cells by using a specific radioactive precursor of heme synthesis. This is accomplished by first incubating the cells with C14-labeled 5-immunolevulinic acid, which will be incorporated into heme. The second step is to collect the cells by scraping, and then to lyse them using heme extraction buffer. Next, diethyl ether is added to extract heme, and the ether phase is collected. The final step is to wash the ether phase with hydrochloric acid to separate porphyrins from heme. Ultimately, levels of heme synthesis can be measured in cell lines, and the results show the differential levels of heme synthesis in various cancer cells and HEK293 T cells.
The main advantage of this technique over the existing methods, like the use of C14 glycine or iron 59, is that C14-ALA is very specific and measures the flux of the whole pathway. Additionally, C14 radiation is very weak compared to iron 59.
The implication of this technique extend toward diagnosis and therapy of cancer and neurological diseases, because altered heme metabolism is associated with various diseases including cancer and neurological diseases.
The seeding confluency for cells for the heme assay depends on the cell type and their growth rate. Seed cells in 3.5 centimeter plates so that the confluency is 80 to 90% on the day of heme measurement. The use of individual plates is recommended.